All right, let's dive into the microscopic world that's running your body right now. We're talking about proteins, these incredible little machines, the absolute workhorses inside our cells that are responsible for, well, pretty much everything.
And that's not just some poetic line. It's the literal truth. Proteins are the foundation of what we are. From the stuff that makes up your hair to the antibodies fighting off a cold, it all comes back to these amazing molecules.
So to really get a handle on them, we're going to approach this like an architectural project. We'll start with the basic alphabet, figure out how to sort our building blocks, watch them assemble into a 3D shape, see what holds them together, and finally, find out what happens when it all comes tumbling down.
Okay, let's jump right in. Before we can build anything, we have to know what we're working with, and that means we need to talk about amino acids.
Now this is what blows my mind. There are hundreds of thousands of different proteins out there, each with its own special job, but they're all made from a standard set of just 20 amino acids. It's like an alphabet. With just 20 letters, you can write everything from a shopping list to a Shakespearean play. Biology uses its 20 letters to write the entire epic of life.
So here's the basic blueprint for every single one of those letters. You've got a carbon in the middle, an amino group, and a carboxyl group. But the real star of the show, the part that gives each one its unique identity, is that R group. This little side chain is what makes each amino acid different, giving it a specific personality that changes everything.
And those different personalities, they are absolutely crucial. We can actually sort these 20 building blocks based on their properties, and that sorting is what determines how a protein is going to fold itself into a working machine.
Okay, probably the most important property to understand is polarity. See, our cells are basically bags of water. Some amino acids are hydrophobic. They hate water, kind of like oil. Others are polar or hydrophilic. They love it. And this one thing is the main force behind protein folding. The water-hating guys desperately try to bury themselves on the inside of the protein, while the water-loving ones are happy to stay on the outside. That simple game of hide-and-seek is what starts to create these incredibly complex 3D shapes.
Now, these properties don't just affect shape, they also affect where we get them from. And this is where it all gets very personal. Your diet. Our amino acid alphabet is split into two teams. The non-essential ones, our bodies are amazing and could just whip those up on their own. But the essential ones? Nope, we can't make them. We absolutely have to get them from the food we eat. And this number 10 is a big deal. It's the number of essential or semi-essential amino acids we need for our health, especially when we're growing. That's about half our protein alphabet that has to come from our diet. If you're missing even one, it can stop the production of thousands of proteins cold. That's why a balanced diet isn't just a nice idea, it's a biological command.
Okay, so we've got our bricks, and we've sorted them. Now for the really cool part. How do we get from a simple string of amino acids to a fully formed three-dimensional machine? This is where biology's architectural genius really shines.
It all happens in 4 levels. First, you have the primary structure. That's just the unique sequence, the specific order of amino acids in a line. This is the blueprint, and it's written in our DNA. Then parts of that chain start to twist into coils called alpha helices or fold into pleated beta sheets. That's the secondary structure. Next, the whole chain folds up into one big, complex 3D shape. That's the tertiary structure, and this is really where the protein becomes functional. Finally, for some proteins, multiple folded chains have to come together to form the final product. That's the quaternary structure, the finished multi-room building ready for business.
You know, a beautiful building is one thing, but it's not much good if it's going to fall over. So what's the glue? What's the mortar holding all this incredible molecular architecture together?
Well, it really boils down to 2 main types of bonds. You've got these super strong covalent bonds that create the backbone of the protein, and then you have a whole bunch of weaker, non-covalent bonds that all work together to sculpt and hold its final shape.
So think of it like this. The covalent peptide bonds linking the amino acids, that's the mortar. It's strong, stable, and pretty much permanent. But the final 3D shape, that's held together by all those non-covalent bonds. They're more like magnets and static cling. Each one is pretty weak by itself, but when you get hundreds or thousands of them all pulling together, they create an incredibly powerful and stable structure. So that final room shape, the tertiary structure, is held stable by a whole team of forces. You've got hydrogen bonds making little connections all over the place. You've got ionic bonds acting like tiny magnets between charged parts. Of course, you have those hydrophobic interactions, which is a huge driver.
And then, for extra reinforcement, you can get disulfide bonds, which are like strong, covalent staples locking parts of the chain together.
But here's the thing. For all their strength and complexity, these protein structures are actually incredibly delicate. They exist in this perfect, fragile balance. And if you disturb that balance, the whole masterpiece can fall apart.
So what happens when things go wrong? When this carefully built molecular machine just collapses?
Well, the technical term for it is denaturation. It's when a protein loses its complex 3D shape. All those higher levels of structure just fall apart. Now, what's really important to remember is that the primary sequence, the string of amino acids, is still there. But without its specific shape, the protein is useless. It completely loses its function. The building has collapsed into a useless pile of bricks. And a lot of things can cause this collapse. Physical things, like heat, are a huge one. This is exactly why a high fever is so dangerous. It can start to denature the critical proteins in your body. But chemical agents are just as bad. Everything from strong acids and alcohol to heavy metals. It really goes to show just how sensitive proteins are to their environment.
You've actually seen this happen thousands of times. The perfect everyday example of this is cooking an egg. The heat makes the clear, liquidy albumin protein in the egg white unravel and get all tangled up, turning it into that solid white mass. You can't uncook an egg, right? That change is permanent. That's denaturation.
Okay, so let's quickly recap what we've covered. First, proteins are all built from the same 20 amino acids. Second, their structure is built up in 4 levels, from a simple line to a complex machine. Third, that final 3D shape is everything. No shape, no function. And finally, denaturation is what we call it when a protein loses that shape and with it, its job.
And that brings us to one last thought to leave you with. We've talked about how the environment can make a protein collapse. But if our bodies are buildings and proteins are all the functional rooms inside, what happens when the original blueprint, that primary sequence, has a typo in it from the very beginning? What happens when you build with the wrong brick? Well, that's a question that takes you right to the heart of genetics and disease.